So membrane transport is how cells move things into and out of them. It's selective, meaning they get to pick what goes in and what goes out. And it's either done passively, which means it doesn't require any ATP or expenditure of energy, or it's done actively, which does require ATP. Now, the passive processes, even though they don't require ATP, are driven by just diffusion, just the propensity of the universe to go ahead and kind of even things out. A special type of passive transport is called osmosis. And osmosis is the diffusion of just water instead of the solutes or the small things that are dissolved in the water. Now, if a membrane is lipid, which they are, and the molecule that we're looking at to move into or out of the cell is also lipid-based, then we can do simple diffusion. So simple diffusion is where the molecule is a lipid, the membrane's a lipid, like dissolves like, and so the molecule can just basically dissolve right through the membrane like it's not even there. Now, the molecule being lipid allows it to move through the membrane, but the concentration gradient between the two sides of the membrane are what are going to drive the movement. Facilitated diffusion is similar, except now we're looking at things that are non-lipid, things that are non-polar or hydrophilic or have a charge, all the same thing. These things cannot cross the membrane easily. We've got that hydrophobic center in the middle of our lipid membrane, and so things that have a charge or that are polar really don't like to cross that area. And so we're going to have to facilitate their transport. We're going to use proteins to do that. So again, we're still using diffusion to drive the movement here. It's a difference between the concentration gradients inside and outside of the cell, and we're always going to move high to low. In this case, we're going to have to move, instead of just randomly through the membrane wherever we would like, such as simple diffusion, we're going to have to take a specific protein, kind of like a little doorway, to go into or out of the cell. If the little protein has a straight channel through it, and we call that a channel protein or channel-mediated facilitated diffusion. Some of the proteins, though, instead of having a channel, they have a kind of a unique shape where they're open to either the inside or outside of the cell. And then as the molecule in question kind of docks with that protein, the protein will change shape and open to the other side of the membrane. So it's similar. We're still using a protein. It's still a type of facilitated transport, and we're still using diffusion because it doesn't require energy. We're still using the concentration gradients between the inside and outside of the cell, with the movement always occurring from high concentration to low concentration. Now, active processes are going to require an expenditure of cellular energy in the form of ATP, but this lets us do some different things. This allows us to move things against their concentration gradient, and go from a low to a high concentration if we wish, but it's going to take work and energy to do that. If the membrane simply grabs a hold of a molecule and shoves it out to the other side of the cell, that is what we call primary active transport. Secondary active transport is where we do primary active transport previously, and that's going to allow us to do some diffusion on the other side. So it's kind of coupling primary active transport along with a type of passive diffusion. And when we do this, we can either transport the two molecules in question to the same side of the membrane, which would be a symport, or we can transport them in opposite directions, which would be an antiport. Finally, we've got vesicular transport. If we need to move a lot of molecules or very large molecules into or out of the cell membrane, then we're going to have to use a vesicle. And a vesicle is basically just a little tiny bubble of cell membrane material. It's a phospholipid bilayer. If that little bubble of material is moving into the cell, that's called endocytosis. And if the little bubble of material is leaving the cell, then we're going to call that exocytosis. Exocytosis is pretty straightforward. Endocytosis has a couple of subdivisions. One is called phagocytosis, where a cell will actually form a membrane around an object and engulf it and it literally eat it. Our white blood cells do this when they chow down on bacteria. Another type of endocytosis would be penocytosis, which is gulping or drinking. So if there's a lot of fluid on one side of a cell and it needs to get a bunch of fluid through quickly, it's going to do a lot of vesicular transport and bring that fluid into the cell. And finally, we've got what's called receptor-mediated endocytosis. So this is where there are special docking proteins on the outside of the membrane. And when these are activated by different molecules, that's going to cause the membrane underneath them to kind of collapse and form a little vesicle that will be drawn into the center of the cell. So remember, the plasma membrane serves as a physical barrier between the cell and basically the rest of the universe. It regulates the movement in and out of the cell, establishes an electrochemical gradient, which we're going to go into in detail in the next couple of weeks, helps with cell communication, and it's critical to transport. Remember, your cell membranes are not passive. They don't just sit there as a boundary to the cell. They're extremely dynamically active. And to move things in and out, again, we've got the two processes, active and passive. The passive processes do not require energy. They all depend on concentration gradients, moving substances from a high concentration to a low concentration. The active processes all require energy. So diffusion is going to be the net movement of a substance from an area of greater concentration to an area of lesser concentration. So imagine, if you will, you're in a large empty room and you take some lemon-scented air freshener and blast a bunch of it into one corner of the room. Now, the concentration of lemon-scented molecules is going to be really high in that particular corner. And if you look at the opposite side of the room, it's going to be really, really low. But over time, the random Brownian movement of the air molecules kind of bonking into each other and bonking into the little lemon-scented molecules are going to start to spread those lemon-scented molecules out throughout the room. If we wait long enough, then the concentration or the disbursement of those lemon-scented molecules is going to be pretty much the same anywhere you look. It's all going to be equal. So diffusion is when we're going to move a substance from an area of high concentration to an area of low concentration until equilibrium is reached. You're not going to have that cloud of lemon-scented air move over to the other side of the room and concentrate on the other side and have the original corner not smell of anything at all. It's not going to do that. It's going to spread out fairly evenly. Now, if you look at a snapshot of the room and look at the lemon-scented molecules, they're pretty much evenly distributed, but they're actually moving around. So if you follow a single particular molecule, it might meander from one side of the room to the other. So everything is constantly in motion. But if you take a snapshot, things are going to be pretty much equally distributed throughout the region. So that's diffusion. Another way to think about it is if you put a drop of food coloring in a glass of water. The instant you put that food coloring in, the concentration of food coloring in that particular part of the glass is going to be very high, with the bottom of the glass being very low. But over time, the little molecules of food coloring are going to get bounced around by, again, the random Brownian movement of the water molecules, and it's going to be evenly distributed throughout the entire glass. Now, the rate of diffusion, how quickly it spreads out, is going to depend on how much difference we have between our concentrations, or the steepness of the gradient is another way to say it, and also temperature. Since the diffusion is driven by the random movement of these little air or water molecules bonking into each other, if we add heat to the system, we're adding energy, kinetic energy, and they're going to bonk around a lot faster, kind of like little billiard balls hitting each other faster, and that's going to increase how fast we're going to reach equilibrium. So here's our example of diffusion. We put some dye into water. You can see that right here, our concentration is really high. And at this moment in time, our concentration down toward the bottom of the glass is going to be really low. But as the water molecules bonk into each other and move around, they're going to start to spread the dye molecules around. And here we have in the center the process of diffusion going on until we reach equilibrium. Now remember, if we look at an individual molecule, it's not frozen right here. It's still moving around. It's moving randomly. It's doing bumper cars with all the other little molecules. But if we take a snapshot, just as we did here with the picture, we're going to find that all of the molecules are fairly evenly distributed throughout the glass. So simple diffusion is where the molecules can pass through the physical barrier of our phospholipid membrane very easily. It means they're going to be small and they're going to be non-polar. They're not going to have any kind of charge on them. So things like oxygen and carbon dioxide, some fatty acids, ethanol, urea, all are what we call lipid soluble. They'll melt right into the plasma membrane. In this case, the membrane cannot regulate the molecule transport. Since these things can go right through the membrane wall without any kind of doorway or help, how much is inside or outside of the cell is all governed by simple diffusion. So here's an example. Here on the outside, we have our interstitial fluid, which is outside of the cell. On the inside, we have our cytosol. And you can see here, we've got a much higher concentration of oxygen outside the cell than we do inside. And so the net movement is going to cause oxygen molecules to simply diffuse through the phospholipid membrane into the cell. On the other hand, carbon dioxide is also lipid soluble. The concentration tends to be higher inside the cell than outside, and so that's going to drive the net movement of carbon dioxide molecules outside the cell. Again, simple diffusion. Now, facilitated diffusion, remember, requires some help. We're going to move a molecule that is polar, that has a charge. And remember, polar and nonpolar things really don't get along very well. And so these polar molecules are not going to be allowed to just simply waft on through the membrane like it's not even there. They're going to have to take a very specific doorway. And so those doorways are proteins embedded within the cell membrane. We're going to call these either channel proteins or carrier proteins. Channel-mediated diffusion is going to move small ions, which have a charge, through water-filled protein channels. Now the channels are subdivided into either what are called leak channels that are just kind of continuously open, and because they're open, they're unguarded, again, stuff can just move in or out of the cell, and that's just driven by diffusion, but because we're using a protein to do this, it's facilitated diffusion. Gated channels are channels, but they're usually closed. Just like the name says, they've got a little gate on them that we can open or close according to different signals in the body. And this is really, really important for when we start talking about how the brain works and how muscles work. We're going to talk a lot about gated channels and what triggers them to open and what moves through them. But what's causing the molecules to move from one side to the other, again, is just going to be a concentration gradient, just diffusion. But what allows them to go through is going to be these protein channels. So here's a little example. Here we have some sodium. It has a positive charge, so it is polar. It is not going to want to go through this hydrophobic non-polar region here in the center of our phospholipid bilayer. It can't do it. So it's got to use a little doorway here, and we've got a little sodium channel. Now you notice this channel does not have a gate on either side, so it is a leak channel. So we're going to go from, again, our area of high concentration to an area of low concentration. Here we've got potassium, again, another positively charged ion, doing the same thing. We've got a potassium leak channel over on the right side, and we're going to move from our area of high concentration inside of the cell to an area of low concentration outside of the cell. Sometimes our protein structures that allow us to do facilitated diffusion are not channels. Instead, they're going to be carrier proteins. The carrier proteins, as I mentioned, tend to change shape to allow molecules or ions inside or outside of the cell. If we're only moving one atom or molecule at a time, it's called a uniport. For example, here is glucose. It's kind of a bigger molecule, and it has a little charge on it, so it's not going to want to diffuse through the membrane. It has to use either a channel or, in this case, a carrier protein. And you'll notice this carrier protein is kind of open here to the outside of the cell, but it's currently closed to the inside. And there's a little space right in the middle here where the glucose fits. So as soon as the glucose kind of plugs into there, then the carrier changes shape. It closes to the outside and opens to the inside. And the glucose can detach and wander off inside of the cell. And then that carrier protein is going to go right back to its original shape and allow more glucose to slowly move its way in.